So in this section of Chapter 5, we're going to look at how your body actually uses DNA. So we've talked about DNA contains genes, and those genes make proteins that do things in the body. So we want to look at what's called the central dogma of molecular biology. The central dogma says that DNA in your body goes through the process of transcription, or is transcribed, to make something called messenger RNA. That messenger RNA is translated into protein. So we go through DNA transcription, messenger RNA translation into protein. And that's the central dogma of molecular biology. Basically how it works, down here we have a cell, and inside the purple area is the nucleus, and this blue shaded area is the cytoplasm, so just outside the nucleus. The DNA in the nucleus is copied into messenger RNA. That's done by enzymes, other proteins inside the nucleus. This enzyme is RNA polymerase, so it's going to copy or transcribe. So when you take notes in class and you're writing down what's on the chalkboard or the projector, you're transcribing. So you're copying that information down. So transcribing or transcription, we're copying the DNA, just a small segment, a gene. The gene goes from here to here, a gene in the DNA, into messenger RNA. That messenger RNA then, this yellow line, that messenger RNA leaves the nucleus and goes out into the cytoplasm, where it joins a ribosome. And we've looked at ribosomes before. We've seen those in prokaryotic cells when they were just floating loose in things like bacteria. And we've also looked at them in eukaryotic cells. It was ribosomes attached to the endoplasmic reticulum that make it rough, endoplasmic reticulum. And we know that rough ER makes protein. That's because it's covered with ribosomes. So this ribosome will attach to our messenger RNA and read it. So it'll start at one end and start reading that piece of RNA and using it to build a protein. And DNA and RNA are similar in structure, and we'll look at that as we go through this chapter, but protein is a completely different molecular structure. A great way to think of the central dogma, if you think of cooking, maybe grandma has a really big cookbook, and if you want to make a recipe, you probably don't want to drag that big cookbook around with all the recipes. You just copy one recipe out. So in this case, we open grandma's cookbook to one page, equivalent to one gene in our genome, and we copy out the information to make grandma's cookies. So that note card with that one recipe would be the equivalent of our messenger RNA. Now that we have our one recipe, we can go into the kitchen. So our cytoplasm out here is our kitchen, and we can read the recipe and make cookies. In this case, you are acting as the ribosome. So you're reading the recipe and transferring the language of ink and paper into the butter, milk, and cream, things that you need to make cookies. At the end, you get a functional product, which would be your protein or your cookies. You wouldn't want to just eat the recipe card, and the cells simply can't just use the messenger RNA. They have to make the final product. So we'll look at some of these steps in a little bit more detail. Make sure that you have them in your notes. Transcription, again, we copy the DNA into RNA. So we're transcribing, making a copy of it. This is where it occurs in our cells. And what's happening inside is that DNA strand, that double helix, is opening or unzipping just a little bit and making a copy using the pairs between A's and G's, C's, and in this case U's. So in DNA we saw that A paired with T and C pairs with G. Well, RNA that we're using for our messenger, it doesn't have the T. Instead, that's replaced with uracil, a U. So uracil replaces thymine, so RNA has a U, CG pairs that are built up to form that message molecule. And RNA is single-stranded, so DNA has two sides, RNA just has that one side. Then we go on into translation, and this is outside of our nucleus. That messenger RNA leaves the nucleus and goes into the cytoplasm, and this is where we actually use the information from the RNA to make a protein, so a functional product. And this blue in the background represents our ribosome, and that's where it's happening. We have another type of RNA called transfer RNA or tRNA, and that's this kind of yellow shape here that's attached to the balls representing our amino acids. And these transfer RNAs come in, and every 3 letters on our message will line up or match 3 letters on the transfer RNA, and it's carrying the correct amino acids. So each of these 3 letters is like a word describing how to build a protein. So we stack them all together one after another, and we make our functional protein once we've connected all of these amino acids. So to make a protein, you stack together a series of amino acid molecules. This is the codon table, and this is universal. So I mentioned you could take DNA out of E. coli and put it into a strawberry and it would work, or take DNA out of a tree and put it into you and it would work most of the time for specific types of genes. Well, the reason it can work is that almost all organisms use the exact same kind of 3-letter vocabulary. So this up here represents those 3 letters in our messenger RNA. The first letter, if it's G, A, C, or U, you can look it up on the chart going across this way. The second letter, whether it's U, C, A, or G, you look it up across the top. And the third letter is listed down the side. So for glutamine, to bring in glutamine, you can either have G, A, or G, A, G. Both of those code for glutamine, and every organism would use that same set of letters or bases on their messenger RNA to put glutamine into a growing protein. And this just shows the process kind of a little more up close. So we have our different locations within our ribosome, bring in methionine, cysteine, serine, valine. So mutations are anything that changes the sequence of the DNA. And mutations can have an effect on the genotype because they're changing the sequence of the DNA, or what's actually said in the DNA. And that in turn affects the phenotype because the genotype determines the phenotype in an organism. Those phenotypic changes could be good, beneficial for the organism. They could be bad for the organism, or they could be completely neutral, not really do much of anything for the organism either way. An example of a mutation, this is a normal fruit fly eye. We can see that's this huge red structure. There's a mutation called bar eye that gives them substantially smaller bar-shaped eyes. And that's obviously for the fruit flies a bad mutation. And again, that's caused by a change in the DNA. There are different types of mutations. There are point mutations that occur in just one base. So if you remember our codon chart from the last section of the chapter, if we change some of these letters, so if we take GAC and change that to GAA, we've changed the amino acid. If that causes a problem with the protein and the protein doesn't function properly, we'll get a different phenotype. So it all goes back to this codon chart and what is actually put into a functional protein, if it's the right sequence of amino acids or not. So that's a point mutation. We'll look more at those on the next slide too. And there are also chromosomal aberrations, so large problems with entire chromosomes or large chunks of DNA having problems. Here's some illustrations that show we have our normal DNA strand up here, and in our normal DNA strand we get a 3-letter match going all the way down, we get our messenger RNA, and we end up with our normal protein. If we do a substitution of one nucleotide, so we trade in one nucleotide for another, what that's going to do at that single location is put in potentially a different amino acid. That could have a huge effect on how the protein actually works. Or it might have no effect whatsoever. It kind of depends on the importance of that individual part of the protein. If we insert one base, then what that does is shifts everything kind of off from there on down, so that not only affects that particular amino acid, but everything else beyond that gets changed. Same thing would happen if we remove one base. You can think of it as a sentence. So if I have a sentence composed all of 3-letter words, so Sam the cat ate Joe rat. So everybody can understand that, right? Sam the cat ate Joe rat. Fairly straightforward. If I change one letter, so maybe change this T to an F, Sam cat ate Joe rat, that messes it up a little bit. We could probably still figure out the meaning of that sentence. However, if I delete the F, considering the fact that we still have to read from beginning to end every 3 letters, I end up with Sam, he, at, or don't know how to say that. That changes the meaning completely. So this is gibberish. Same thing would happen in the DNA. If we insert something, everything after that could just be gibberish. If we add an extra letter, so do the same thing, Sam, and now we're just going to add a random letter, and that changes, so that's our 3-letter word. And again, everything past that extra letter is just gibberish in our DNA. So there's different ways that the DNA can have mutations. Depending on what type of mutation it is, some of them might not cause a problem at all. Others will be substantial problems. So those are point mutations. We can also have chromosomal aberrations, so huge problems within our chromosome. So here's our normal chromosome. We've got gene 1 is purple, gene 2 is blue, and gene 3 is green. If we have a gene deletion, maybe in this case gene 2 gets lost, so that changes it because we no longer even have the appropriate information for gene 2. We just have 1 and 3. We could relocate a gene. Maybe gene 1 gets shifted and moved to a different location. That could be a problem for the organism because usually right next to each of the genes there are areas that regulate the genes, tell the body how often to use that gene. If a gene gets moved and separated from its regulation, it might not be functioning correctly. We can also duplicate a gene. So in this case, our gene 3 was duplicated. Now we might have too much of a particular protein, and it could cause problems within the body. So those are all different types of mutations that could occur. Those mutations could be spontaneous, so sometimes there are just mistakes that happen in the DNA. So when the DNA is copied, it could just have a spontaneous mutation, an accidental error could occur. And that happens with a regular rate, so we know about what the rate of spontaneous mutations are. A couple things that can induce or cause mutations on their own are radiation. So radiation induced, this is why when you go to places like the dentist, this gal here is wearing this huge lead collar and cape to protect her body from the x-rays that'll be coming out of this machine to get an x-ray of her teeth. Another type of radiation comes from the sun. So this gal here has a pretty bad sunburn. It's all bright red. And sunburn is caused by UV radiation. And what happens with UV radiation, it actually hits your skin and goes right into the cell and hits the DNA. So here's a strand of DNA, and any place in your DNA that you have, especially thymines, close together, when it strikes against those, it's at the right wavelength of light hitting those thymines that it causes them to lose the connection across and form a bond between the two thymines. Really, what that does in the DNA then, it forms a little kink or distortion, actually misshapes your DNA right there at that point where you have that cross-linkage. Now, there are enzymes in your body, and their sole job is to constantly scan and monitor your DNA, look for this problem, and the enzyme fixes it, which is why even though all of you have been exposed to sunlight, and some of you may have even had sunburns before, you all don't have skin cancer because that enzyme fixes it. The problem occurs when you get a lot of damage that these enzyme fixers can't keep up with it, or if you have a mutation in that enzyme. So some people are allergic to the sun. They simply cannot go out in the sun, and you see them wearing like big almost spacesuits when they go outside, or they only play outside at night. And the reason that they can't go out in the sun is that every time they're exposed to UV light, there's a high probability that that's going to cause a mutation, which can lead to skin cancer. So sometimes you see little kids that have this particular enzyme problem, and they've had skin cancer removed 5 times, and their moms run around with little light meters checking everywhere the child goes to keep them out of UV light. So that's one type of radiation induced. Also, chemicals can cause mutations. Lots of chemicals purposefully or unintentionally put into the environment. Cigarette smoke is one of those. So lots of chemicals in cigarette smoke, not the tobacco itself, which is what smokers are after, is that the tobacco, but all of the other chemicals that are put in there. And those other chemicals are there mostly to make it more addictive and give it different properties. But those other chemicals can induce mutations. In fact, they do several things. They shut down the cilia in your lungs, so your lungs no longer clean themselves properly, which allows chemicals to build up there. Also, because you're breathing into your lungs, that delivers the chemicals directly to your bloodstream. So people who smoke may not just get lung cancer because those chemicals get in your blood and travel throughout the body and can give you various types of cancer around your body. Some of those chemicals in cigarettes actually interfere with other enzymes that will normally monitor your DNA. Very similar to our enzyme for UV light, there are enzymes. There's one called the guardian angel gene. And that guardian angel gene just constantly monitors your DNA for problems. Well, some chemicals in cigarette smoke actively shut down that guardian angel gene, and so that increases the likelihood of getting cancer because you no longer have the enzymes there to look for problems and fix it. Changes in DNA can lead to sickness, and these are changes that are occurring in the egg or sperm and passed to the offspring, or that have been carried in the parents too and passed to the offspring. So we've already discussed Tay-Sachs disease when we went over pedigrees, so that's an obvious example of a problem in the DNA. Another one, and there are lots of these, another one is fast flush reaction. So people who have fast flush reaction, there's a problem with one of their enzymes. Normally when people drink alcohol, the body will take the alcohol and break it down and turn it into a non-toxic substance. However, people with fast flush reaction, partway through the metabolism of alcohol, one of those enzymes doesn't work correctly, and so it builds up a toxic molecule for a period of time in the body. So immediately when they drink alcohol, they turn bright red, their face gets flushed, and they never get kind of a happy buzz from the alcohol. Instead, they go right straight into feeling sick from the alcohol, just because that toxic product builds up. So that's another change in the DNA that can lead to sickness depending on environmental factors. And again, we've talked about Tay-Sachs, but this enzyme gives an example of either Tay-Sachs or fast flush, basically something wrong with the enzyme and you end up with a toxic product.